Hello everyone, this is Shuli from Group 1. Today I'm going to talk about animal cloning and how can cloning help and hinder medical research. Animal cloning has been a controversial issue for a long time. Many people have ethical and moral concerns about animal cloning. According to recent surveys, 64% of Americans think cloning is morally wrong. 63% are not willing to consume cloned food. Cloning promotes the objectification and commodification of animals, treating these living things as mere machines for human manufacture. In addition, cloning highly productive animals will make animal welfare problems become intensive. Because the cloning of animals involves great suffering. Problems occur in cloning far more often than with any other method of reproduction include large offspring syndrome. This occurs in over 50% of clones. Hydrops, the condition that animal swells with blood. This occurs in 28% of clones. A high rate of late-term pregnancy loss, pregnancy complications, painful labor, and surgical intervention is unique to clone pregnancies. For example, a cloned embryo has to be implanted into a surrogate mother, but cloned embryos are often large and can result in painful births. Many clones even die during pregnancy or birth. Majority of those that survive also die in the early days and weeks of life, from problems such as heart, liver, and kidney failures. Recent data on the success rate of cloning shows that there are high rates of miscarriage, stillbirth, early death, genetically abnormalities, and chronic diseases among cloned animals. The efficiency of animal cloning has typically been about 1 to 2% only. That means for every 100 embryos, 98 of them fail to produce a living animal offspring. Some people may think that cloning can help medical research. For example, genetically modified animal cells and organs can be transplanted into humans to reduce or even eliminate rejection of the graft. There are also some other techniques using cloning which seem to benefit. But the potential dangers of cloning can be very serious. Most commonly happen is mutation of genes. In cloning, the transferred nucleus does not have the same program as natural embryo. What scientists do is just reprogram the nucleus. Complete reprogramming is needed for normal development. But incomplete reprogramming will cause the embryo to develop abnormally or even fail. Besides, clones show difference in telomere length, which will affect animals' lifespan. As cells divide, their chromosomes get shorter. This is because the DNA sequence at both ends of chromosome called telomeres shrink in length every time the DNA is copied. The older the animal is, the shorter its telomeres will be, because the cells have divided many times. This is a natural part of aging. When scientists looked at the telomere length of cloned animals, they found chromosomes from cloned cattle or maize had longer telomeres than normal. These cells showed signs of youth and seemed to have an extended lifespan compared with cells from a natural cow. On the other hand, Dolly the sheep's chromosome had shorter telomere length than normal. This means that Dolly's cells were aging faster than the cells from a normal sheep. All this showed that techniques of cloning is not mature. In conclusion, cloning is not that safe yet. It is really risky to clone because of unknown long-term effects. And it's also not ethical to clone. For reasons above, I think cloning should be banned. Thank you very much.